This is 5 articles this video is introducing on the subject of the surprisingly complex tRNA subsystem. tRNAs serve as a linker molecules to translate mRNA sequences into protein sequences. But these require extensive processing by pre-existing protein-based molecule machines. Essentially, what these 5 articles point out is just by looking in detail at one aspect of what's going on within the cell, you begin to understand the sheer complexity of the whole process. And so the writer here is just looking in detail over 5 papers, and they're long papers, at just one aspect of the process, and this is tRNAs. If you were to drill down to the same level of detail in any other aspect of the cell, you would have the same level of complexity. The overwhelming conclusion from such detailed analysis is that we now can come to the convinced conclusion that the cell is far too complicated to come about by any process other than incredible intelligent design. It works so well, and it is so complicated that this can only be the product of intelligent design. Furthermore, there is no evidence of large-scale junk DNA or any other evolutionary baggage. Such evidence as is brought forward tends to be very small scale and can be probably explained by other methods, or can be the reasonable expectation of a few thousand years of genetic mistakes, but not what you'd expect from millions of years of genetic mistakes being cobbled together to produce some sort of design by a process called natural selection, which is not particularly known for its finesse and its ability to do detailed changes. Natural selection, by and large, deals with big problems, not small-scale problems. The fact of the matter is, wherever we see such changes, such as the beak sizes for the finches in the Galapagos Islands, these are not the product of any evolutionary process that's random. It looks like they're the product of a pre-design ability in the genetic code to change according to the environment. And that is a design feature. That's not a product of evolution. In fact, mutations and mistakes will always degrade a design system. It won't improve it. It's called genetic entropy, and that's the work of John Sanford. So these 5 papers just give us an opportunity to delve down in detail in one aspect of the cell and come away utterly amazed at God's incredible design. And truly, the right response to modern biology is not to say how wonderful evolution is. It is to say how marvelous is the Lord and God of all creation. And you see, the Bible frequently makes us want to point to God as the creator of the heavens and the earth, and to glorify him. For that's the appropriate response of the human heart, is to give thanks and glory unto God for his creation, which, even though fallen from the glory that it once had, is still incredible. Well, thank you for listening.